All right, good afternoon, everybody. Welcome to Wednesday afternoon of Chem 170 with your host, me, Dr. White. All right, if you haven't noticed, I worked real hard last night and this morning. And this morning I posted test number 3 scores. Later today or this evening, by tomorrow morning, I will send out email with your individual points for each question on, or each of your answers on test number 3. Tomorrow in class, I'll go through the answers for test number 3, but I'll cut it out of the video. And if you miss it, always feel free to come to my office hours, like I'll have one tonight. And that's that. All right, some important things. First of all, sometime later today or early tomorrow, I will post test number 4 information. It's this summer, it gets quick and faster in the end, and it is. Next Monday, I'll give test number 4, just like I did 3. At the end of class, I'll send an email with the password for test number 4, and you'll have until Tuesday, say 5 p.m. to do test number 4. And then I'll hopefully get it graded by Wednesday morning and go through it or Tuesday, Wednesday morning, and Wednesday in class, I'll go through it. Make sure you're not late. Also, that's correct, Jimmy. We just have test number 4. And I was just about to mention it, so you're reading my mind. Oh no, I better put some tin foil here. Not. But anyways, a week from today will be the final exam. The final exam will be cumulative, the whole semester. The best way to study for the final exam is, one, come to my lectures next week, because Monday, Tuesday, and Wednesday, what I will do is go a whole semester review over those 2.5, 3 days. And that always helps my students when I do the last week, like next week will be the last week review for the final. Also, if we were face-to-face, even though you're online, I'm doing the same thing. I guess there's a way I could make sure it didn't happen, but I don't. But anyways, use your hourly exams as practice problems. Use your hourly exams as practice problems for the final. Why? Well, look at my practice problems all semester. Were they identical to what's on the test? No. But were they similar? Yes. I like that. Yes. Hold on. No, yes. It's awful humor Wednesday. But those will help you practice for the final. I see a message. All right, Jimmy, this Monday will be test number 4. I'll send out an email later today or tomorrow morning. And so again, let me repeat. This Monday will be test number 4. This Thursday will be the final exam. Now on Thursday, we'll have no lecture, because when I have the final exam face-to-face, I give it, you're done, we leave. Well, there'll be no lecture. So next Thursday, starting at 2 p.m., I will send out the email with the password for the final exam. And it'll be just like the others. You'll download a PDF file and you'll make a PDF file of your answers. And I'll give you until this next Thursday, let's say 11 a.m. Friday, because I gotta get the grades and I've got a deadline. So 11 a.m. Friday, if there's a problem with that, let me know and we'll talk about it and see if I can help you out. Any questions? Oop, I see some chat stuff. All right. All right, here's what I'm going to do today. First of all, as promised, today I'm going to go through the lipids, otherwise known as fats and oils problem set. we'll finish up the carbohydrates chapter, and then I'll go into new stuff, which will not be on test number 4, but there'll be some points on the final. And then we'll see how much time. We'll do a little more practice for the carbohydrates. And tomorrow we'll do, first of all, the carbohydrates problem set. Then after that, I'll do some more practice of fats and oils and carbohydrates, things you should know for test number 4. So either watch the video or come to class tomorrow. Also tomorrow, I'll go through, like I said, test number 3 answers. All right, I think we're up to date. Any questions? All you out there, I'm listening or looking. All right, let's get to work. All right, thumbs up, people. Do you see lipids practice problem on your screen? Oh, by the way, Dr. White got really lazy at the end of the semester. What's a lipid? C knows. And a lipid is a chemical or substance that's obtained from something that was living, plants and animals and people, that is not soluble in water, but is soluble in non-polar solvents. All right, name one type of lipid. Well, a fat is a lipid, a oil is a lipid, a steroid is a lipid. So I have, just to remind you, remember there are 2 types of lipids, and saponifiable lipids are fats and oils. Non-saponifiable lipids are steroids. I'll say that again in slow motion. There are 2 types of lipids. One type, saponifiable, that's a fat or an oil. The other type of lipid, non-saponifiable, are steroids, your hormones. Now, what's the difference between a fat and an oil? Hint, you should know this. A fat is a solid or semi-solid at room temperature. An oil is a liquid at room temperature. What's yellow grease? Pig's fat? I don't have it there. Oh, excuse me. You should know. What's white grease? Pig's fat. And what's tallow? Beef fat. And Dr. White worked with all those in large quantities when I worked for a company that at that time was called Unichem Chemicals, located 47th and Grace in Chicago. All right, let's take a look at some problems. Give the organic products for the following. So what do we have? Well, look for what's different. Oh, an ester, another ester, another ester, a double bond. Well, double bonds don't react with sodium hydroxide and water, but esters do. You get back to the alcohol plus the carboxylate anion, the carboxylic acid you would have used to make that ester. I've got 3 R groups here. Therefore, you get 3 carboxylate anions. And for each ester, you get this alcohol put together glycerin. Same pseudo fat, pseudo meaning it's not really a real fat, but just to give you a practice. And that is a triglyceride, triester, not 1, not 2, but 3. Here they are, acid and water. Now, under these conditions, double bond won't react, but the ester will. And you learn for test number 3. And by the way, everybody, just about everybody did really good on test 3, which made me very happy. Thank you. And here you have a ester, acid, and water, and you get back the alcohol and the carboxylic acid you would have used to make that ester. Ester 1, you get this alcohol and this carboxylic acid. You don't lose the double bond. Remember, do you break carbon-carbon single bonds? No, don't. You'll be wrong. Next ester, you get this alcohol plus this carboxylic acid, and the final ester, you get this alcohol plus this carboxylic acid. Now, here I have hydrogenation. This molecule with hydrogen and nickel catalyst. Esters do not react with hydrogen. Esters do not react with hydrogen. but double bonds do. You learned that for test number 1. Ooh, I like that. Ding, 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 ding. It's awful, really awful humor Wednesday. And you break the pi bond, each carbon gets a hydrogen. So the only thing that changes here is this double bond. And notice, in case you couldn't see it, I have look, and I broke the pi bond, each carbon gets a hydrogen. And if you change the catalyst, same thing happens. Now, if we look at E, what do we have? We have an triglyceride steam. What's steam? Hot water. Remember, water can hydrolyze an ester to give you back to carboxylic acid plus the alcohol you would have used to make that. But normally we use acid catalyst. Another way to get a reaction going quicker is raise the temperature. And hot water will do that, which is what steam is. In a plant, we use high pressure steam, which is really dangerous stuff to work with if you're not careful. It can kill people. Hold on 1 second. Let's see if I can find something quick. I forgot. You can see high pressure steam, and I think we were up about above 200 degrees C, 240. That's pretty hot. That's about 500 degrees, a little higher than that Fahrenheit. But anyways, let's get back to work. Oop, wrong thing. Oh, I know what I got to do. That thing. All right. So here we have our 3 esters. I get this alcohol plus this carboxylic acid. Does the R group change? No, you're not going to affect that double bond. This one, do you break carbon-carbon single bonds? No. So you get this alcohol and this carboxylic acid. And finally, the last one, notice has 1, 2, 3, 4. The carboxylic acid has 1, 2, 3, 4. Oh, I got it right. And you get this alcohol, and you get the glycerin plus the different fatty acids. Ooh, yellow grease plus acid and water. You should know for both yellow grease, white grease, and tallow, this is the representation. R sub S is chiral group, which I talked about, R sub O, oleal. And what happens? Well, acid and water, ester, you got back the alcohol and the carboxylic acid you would have used to make that. The first ester, you get this alcohol, part of glycerin, plus stearic acid. That's what the R sub S. Now, if on a test, I ask you to draw the structure of stearic acid, do not put down R sub S. It's only when you see it in a reaction do you use that abbreviation. If I ask for a structure, show the carbons. All right, second ester, you get this alcohol. What's the R group? R sub O, so you get oleic acid. Final ester, guess what? You get this alcohol, so you get glycerin, and you get stearic acid again, but I ask, give the products, not balance it. If you want to put 2 in front of there, that's okay. I don't. I'm a lazy organic chemist, and we don't balance equations unless we have to use it to figure out charge weights, how much chemicals to react to make something, and you don't in my class. All right, tallow, use the same structure, and you get the same products. White grease, use the same structure, you get the same products. Now, one thing I forgot to tell you something. It's an inside joke from Dr. White. Notice I have R sub S, R sub O, R sub S. What does letters stand for? SOS, save our ship, help. That was my joke, inside joke. All right, now, here we have yellow grease with steam. Remember, steam is hot water. You get back the carboxylic acid and the alcohol you would have used to make that ester. Well, if we come up here, it's the same products as acid catalyst, glycerin, stearic acid, oleic acid. Same thing for tallow. Now here I have yellow grease reacting with sodium hydroxide and water, saponification of an ester. And therefore, take an ester, base and water, sodium hydroxide is the base, you get back the alcohol, and because you're in base, the carboxylic acid becomes the carboxylate anion of the carboxylic acid you would use to make that ester. And in this case, you first ester glycerin and sodium stearate. Second ester, the second hydroxyl group of glycerin and sodium oleate, R sub O. And the third one, you get the third hydroxyl group of glycerin, so you get glycerin, but you get, again, sodium stearate. I didn't put a 2 there. If you wanted to, go right ahead. Give the starting material for the reaction that when you react something with sodium hydroxide and water, you get glycerin, sodium oleate, sodium stearate. And it could have been yellow grease, white grease, or tallow. Now, hidden here, what is a surfactant? And a surfactant is a molecule that interacts at the surfaces of different compounds or surfaces. And most, not all surfactants have a nonpolar tail and a polar head. There are nonpolar surfactants, but most of them are polar. Whoa, we're done with the problem set. Any questions? Any questions? I've got a question. Ooh, let's have some fun. Give the product or products for the following reaction, 3 points each. Everybody, thumbs up if you can see it. Thank you, Jimmy, and everybody else. Just you. But everybody else in YouTube land, hi there. I gotta charge you extra, Jimmy, for having a good time. One other thing I neglected to say, after I go through the test tomorrow, I'll talk about what's your current grade, and next Monday or probably, no, Tuesday, when I send out the scores for test number 4, that Tuesday or Wednesday, I'll tell you how many points you need on the final to get a good grade. And when you're done, give me a thumbs up. All right, let's take a look at this. What is R sub O? That's an allyl group. And let me remind you of this. You should know this is the structure of oleic acid. Remember on a test, I didn't have the problem set, but draw the structure of oleic acid, and that's this. And if I were to take this and react it with hydrogen and a catalyst, let's say nickel, remember double bond, H2 catalyst. I keep on wanting to write and I keep on forgetting. Students don't know cursive anymore. How sad. But anyways, catalyst can be nickel, platinum, or palladium. Oops, palladium. And you break the pi bond and each carbon gets a hydrogen. Now, carboxylic acid, can you hydrogenate that easily? No. So what's going to happen? Do you break carbon-carbon single bonds? No. So I'm going to have this, break the pi bond. Each carbon of the double bond gets a hydrogen. And then the rest of the molecule. Now, if we look at what I just drew, we have, this is really CH2, 7 plus 2 plus, 7 is 16. So I can rewrite this molecule like this. And this is stearic acid, which you also should know the structure of. Now, what is R sub O up here really? This right here. And what's this? This is what I mean by R sub S. So what happens? This becomes this, because I lose the double bond, carbon-carbon. So do esters react with hydrogen and catalyst? And the answer is no. So I'll still have my ester, actually my triester, triglyceride, and each R sub O is changed to R sub S. And that's what they mean by hydrogenated fats and oils. Take something like this with R sub O's, a lot of them, corn oil or vegetable oil, hydrogenate it, you get a hydrogenated oil like this. And this is going to be a liquid at room temperature. This is going to be a solid, semi-solid. Put some yellow coloring, flavoring, water, and you got margarine. You can make a lot of money, and they do. All right. Now, one thing I didn't have on there that you should know, how to describe with words and a general reaction what happens when you eat a fat or oil. And what's the functional group you should know in a fat or oil? And that's an ester. And when you eat something, when I was young, about 2 or 3, my mother used to tell me, chew your food or better, or you're going to choke. And guess what? She was right. I learned that lesson the hard way. Oh, in case you're wondering, I survived. But how do you describe with words what happens after you chew something with fat or oil in it and swallow it and it goes in your stomach? In your stomach, you have acid and water. and that's called acid hydrolysis. I'm spelling it wrong. I can't spell Wednesday. Hang in there. My favorite spell check. I think I spelled it right. Let's find out. No, I was way off. Sorry about that. That's not right. What the hell? I'm going to just do my own way. Now I got it. I can't spell Wednesday. Acid hydrolysis of an ester. Did I ever tell you I was always the first one down to spelling bee? But this part I know, and that's what's the general reaction. Take an ester, react it with acid and water, and you get back the carboxylic acid you would have used to make that ester plus the alcohol you would have used to make that ester. So you should know, leaving my class, important. It's Subtle Tuesday. You should know how to describe what happens in your stomach when you eat a fat or oil. That's acid hydrolysis. Now it looks right to my brain. It's, oh, I can't spell Wednesday. Acid hydrolysis of an ester. And you should know this, and you should know how to describe it with a general reaction. The functional group in a fat or oil is an ester. You have an ester, acid, and water. You get back the carboxylic acid plus the alcohol you would have used to make that ester. Now, one other thing I didn't mention here, or it wasn't on the, hold on one second. I didn't mention on the problem set, but you should know, hold on one sec. All right, Jim, you too, okay. Well, I'm not alone. All right. And leaving my class, you should know how does soap work. And first of all, you should know what's the structure, general structure of soap. And that is soap is a molecule with nonpolar tail and a polar head. And water is polar, and dirt and grease are nonpolar. And the most important piece of the puzzle, how does soap work? And you should know this, is like dissolves like. Notice I went to super large font because it's that important piece of the puzzle. Like dissolves like. Polar things are soluble in polar things. Non-polar things are soluble in non-polar things. And if you mix two together, they're not soluble. Well, water's polar and dirt and grease are non-polar. So water isn't going to get that off your hands or anything else. So what do? You use the soap. And let's look at a magnified piece of dirt on my hand, like maybe if it was right there. This is highly magnified. I can even find dirt that has the word dirt on it. Oh, is that awful humor? Yeah. But anyways, dirt is nonpolar. Use soap and water. The nonpolar tail of the soap is going to be attracted to the dirt, like dissolves like. The polar head wants to be as far away. Well, you're going to use a lot of molecules of soap. I'm just going to show some of them because this surrounds at 360 degrees. And you should know this is now called dirt surrounded by soap like this, a micelle. And now micelle looks polar to water. And water looks at the micelle and says, micelle, you're polar, I'm polar, let's go down the drain together. And they do. So let's go through this again. You should know this. Hint, hold on, let me really stress it. You should really know it. And how does soap work? You should know the structure of soap. Non-polar tail, polar head. Water, polar. Dirt and grease, non-polar. And the most important piece of the puzzle, dun, da, 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 most important piece, like dissolves like. And what that means is when you have a piece of dirt and you use soap and water, the soap non-polar tail lines up like this on the dirt. The polar head wants to be as far away from that. I like that, far away, and it is. And it slowly surrounds the dirt, the form that you should know, a micelle. It looks like a G. Let me fix it up. A micelle. And a micelle looks polar to water. Water looks to micelle and says, I'm polar, you're polar, we know like dissolves like, let's go down the drain together. And they do. All right, any questions on lipids? Oh, one other thing I didn't have there. You should know how to draw the ring system for a steroid. And that's a 6-membered ring, next to it fused, a 6-membered ring, up here, 6-membered ring, and next door here, a 5-membered ring. A, B, C, D. 3, 6, 1, 5, like that. No other way. All your steroids, your hormones, have that in common. All right, I can close this. All right, let's get back to carbohydrates. Now, a molecule you're all familiar with, sucrose, and you should know this. You should know sucrose is your table sugar. You know, the white stuff, powder, not powder, solid, granular solid that you put in your coffee, tea, or whatever else you want to sweeten up. Now, what is sucrose? Sucrose is the cyclic form of glucose bonded to the cyclic form of fructose. And this is an acetal linkage right here that links the two together. So what should you know? All right, class, do you see sucrose that I just wrote on the board? Thank you. All right, you should know sucrose is glucose bonded to fructose. So what happens when you eat some sugar? Well, you have acid hydrolysis of a acetal. And in your stomach, if you have sucrose and you react it with acid in your stomach and water, what does your body make? Glucose and fructose. And conversely, if I take glucose plus fructose and add an acid catalyst, what do you make? Sucrose. And you should know this. Now, one interesting thing, do I have time? I do. A number of years ago, some of the mothers groups were very upset with food companies because they blamed a lot of the maladies their children were having, medical issues, on the fact that a lot of food contained, especially food directed or marketed to children, contain high fructose corn syrup. And they said the fructose in there is causing all these problems, medical problems. Well, the food companies finally said, oh, we hear your pain. And you know what they did? They reduced the fructose level and went back to what they used to have, sucrose. Now, why am I sort of snickering at this? Snicker. Because in your stomach, sucrose makes glucose and you're still making fructose. Now, this is my personal opinion. I label it as such. First of all, the mothers groups were right. There is a problem, but the problem wasn't the fructose itself. It was the high calories of all this food. Trust me, I've been fighting that all my life. Yeah. But anyways, so by changing fructose to sucrose doesn't change the calories, and you're still making a lot of fructose in your stomach from the digestion of sucrose. So that's just an interesting sidelight from Dr. White. All right, you know something? I think now would be a good time to take a 5-minute break. I'm going to do a little and come back at 2:50. All right, everybody take a 5-minute break. I'll see you in 5 minutes. let's get going. All right, let's get back to work. Let's finish up fats and oils. Now, I have here amylose and amylose pectin. Ignore that. Now, one thing I wanted to teach you, one, starch has this structure, and this is a fancy way of drawing the cyclic form of glucose. If you look at the bond right here, the oxygen is bonded from below both rings, below. Now, cellulose, such as paper or cotton, notice the links, one glucose molecule in this polymer and is long, is above the ring, and the other is below. One's called alpha and the other's called beta, and I always forget which is which. But because of this difference in stereochemistry, starch can be hydrolyzed acid and water to make glucose very quickly. Cellulose takes a long time. What does that mean? That means when you eat starch, your stomach, the time it's in your stomach is relatively short, and starch can be broken down. However, if you ate something like grass or hay with cellulose, or worse, paper, it's not going to break down, going to stay in your stomach long enough in your body to break down to give you glucose. However, if you look at cows, and here I'm way beyond my level of animal husbandry, I mean way beyond, but they've got 4 stomachs, and the cellulose, like hay, grass, whatever they're eating, has a longer time to break down to get glucose. Same thing with a horse or an elephant. They've got huge stomachs, and that means it takes a lot longer to break down the hay or cellulose to make glucose to keep them alive, nourishment. Now, look right here, you see the hydroxyl groups on cellulose. How does a towel or a paper towel work? I have it. You know what a towel or paper towel looks. I don't have one handy. Nope, I don't. But anyways, the hydroxyl groups in a towel can hydrogen bond to water. The OH groups in the cellulose in cotton or paper towels are not treated, paper used for printing and writing on has special treatment that allows you to have the ink on there, but they don't absorb moisture that well. But paper towels or cotton towels hydrogen bond to water. Now, have you ever noticed towels don't work as well when they're older? Well, if you've been using a fabric softener, the fabric softener blocks a lot of the hydroxyl groups from hydrogen bonding to the water, which is why they don't absorb as well. In the industry, that's called re-wet. Yes, I've worked with this because I work with fabric softeners. I know about it. I invented a fabric softener that had better re-work than any others. Re-wet. So what do? Well, you wash the towels a couple of times without a fabric softener. That strips it off and it absorbs better. Now, how many of you have heard the term, wow, that cloth or clothes breathed real good? Nope, I don't hear it breathing. Oh, that was awful. What they're talking about when they say how well a shirt or something breathes is how well does it hydrogen bond. The more synthetic fibers, which don't do a good job hydrogen bonding, like if you had all polyester shirt, and back when I was in high school, that was the fashion. Oh, it's like being in a rubber suit. It was miserable. I was glad when that went out of fashion. That shirt, no, so I had a jacket and pants made out of polyester that my mother bought. I think it was senior year of high school. That when it went out of fashion, I didn't even donate it. I didn't want anybody else to be miserable. Right into the garbage dumpster. Yeah, good riddance. But anyways, when you have cotton like this, it hydrogen bonds better, which means it can absorb your sweat, or women, your perspiration. And that's what they mean by breathing. And again, fabric softener, if your clothes don't absorb like they used to breathe, wash it once or twice without a fabric softener. All right, one of the things I wanted to go over again that I went over yesterday that you should know. Oops, wrong color. This is glucose. And it's in equilibrium with what? Remember the reaction. If you have an aldehyde or ketone plus alcohol and acid, test number 2 material. Keep your eye on a carbonyl carbon. What's attached to it is still attached to it. Our primer hydrogen, our double prime, this becomes OH group from the alcohol OR. Hemiacetal or hemiketal. Now, this is intermolecular reaction between two molecules. When they're in the same molecule, and it's this hydroxyl group on carbon C5, where this attack that to form this bond, this will attack this carbonyl, like here. And you should know what the product is. What's not involved in the chemical reaction does not change. So I'm just going to rewrite it. Now, the carbonyl still has a hydrogen. This one. And these are connected in the backbone of the sugar. Now, this oxygen here becomes this one right here. So it's going to be bonded to this carbon that was my carbonyl carbon. It's the world's ugliest, but easy to draw carbon-oxygen bond. And what else will be on that carbon? OH. And you should know, if I give you this structure and equilibrium, how to write this product. If I give you this structure, you should be able to either write glucose or write the actual structure for the starting material. You should know this. All right, the other one you should know And this one I sometimes get wrong, so I'm going to peek. Yep, I thought I got it wrong. You can tell this is an area I didn't work in heavily in industry. Oops, that's not what I wanted to do. This is what I want to do, and here's what I got wrong. And you don't have to know the structures of these like I don't. All right, let's take a look at this. This is fructose, the sweetest sugar in nature. And it's in equilibrium. Same reaction. Ketone or aldehyde, in this case, it's a ketone plus an alcohol and acid catalyst if we're intermolecular, separate molecules. Keep your eye on the carbonyl carbon. What's attached to it is still attached. This becomes an OH, and you have OR. Now, when it's in the same molecule, carbon C5, this is 1, 2, 3, 4, 5. And that also, just like this attacks that, here's my carbonyl, this hydroxyl group attacks that carbon. Now, what's not involved in a chemical reaction, I rewrite. This carbon is this carbon right here, which is this carbon right there. It'll have a hydroxyl group, and I made a mistake. Thank you for erasers. And this is bonded like this. And this bond right there is this bond right there. And you should know, if I give you this structure, you should be able to draw this going this way. If I give you this one, you should be able to draw this or write the word fructose. Now, how can you tell the difference? Glucose forms, and I think it's called a glucose side, the carbon with the long oxygen bond and the hydroxyl group on that carbon is the top carbon. For fructose, glucose, let me do it again. For glucose, this cyclic structure, the glucose side, I think it's called, is the top carbon, has the long bond to oxygen and the hydroxyl group. For fructose, this hemiacetal has the same hydroxyl group on C5, this oxygen, but it goes to, and I did it wrong. This is C2. Oops, didn't want to do that. There we go. This carbon here is this carbon, carbon 2. And the oxygen here is to that carbon. And so this carbon on C2 becomes this one, which is this one. So you should know how to do that. And those are things you should know. Hint. All right, leaving my class, you should know how to describe with words and a general reaction what happens when you eat a carbohydrate. You know, the good stuff, French fries, pizza, potato chips. Hungry, all right? No, I don't need that. Like I need a hole in my head. But what functional group holds glucose together in a carbohydrate? And that's an acetal function. And therefore, how do you describe with words what happens when you eat and chew and swallow a carbohydrate? It's acid. Now it looks right. Hydrolysis of an acetal. And what's the functional group? Acetal. What's the general reaction you should be able to write? And that's an acetal or ketal. You learned this for test number 2. Acid and water. You get back. Keep your eye on the carbon with the 2 oxygens, single bonded. It's rare. This and another reaction are all you ever see or I've seen. And you get the same products. You get back the ketone or aldehyde you would have used to make that acetal or ketal, and you get back the alcohol. Now, it just so happens in glucose, these are in the same molecule. But you should know how to describe with the words and general reaction what happens when you eat a carbohydrate. The key functional group that holds glucose molecules together is an acetal. And how do you describe that with words? That's acid hydrolysis of an acetal. And as you learn for test number 2, take an acetal or ketal, acid and water, you get back the aldehyde or ketone you would have used to make that acetal plus the alcohol. And this you should know leaving my class. Do we have time? We do. Let's talk about some fun things with now, like I said, cellulose is D-glucose with the different linkage, all right? All right, look at cellulose. What does it have on the molecule? Hydroxyl groups on carbon. And what are those? Alcohols. And guess what? I can react alcohols, and people do in cellulose. Now, one of the earliest things that they did was put on a nitro group, and that's called nitrocellulose. And that was used as a polymer for the backing for the original motion pictures from the 1900s, 1910, 1920s, and even a little later. Problem with nitrocellulose, after many years, it gets unstable, not as bad, but sort of like nitroglycerin, and it will combust. And back in about, I think the 70s or 80s, I forgot who started it. There was a major project. All the old films, there's some great ones from the 1910s, 20s, and early 30s, were transferred over to more stable media film. Some of those copies were going up in flames. All right, that's one thing. Now, you can take cellulose and do a strange reaction. Strange because Dr. White doesn't know it that well, because I've never done that. All right, everybody see on the screen cellular cellophane? Now, cellophane, this is a reaction I've never done. Have you ever gotten candy that's in that plastic crinkly paper? Or sometimes you'll get certain food like crackers or stuff in, they don't have a picture here. Well, how do you make cellulose? You take cellophane, react with sodium hydroxide and carbon disulfide. And you make this, I don't know even know what the name of the functional group is there. And they don't have a name of it here either. But anyways, this functional group on there makes that clear, crinkly plastic that they use for candy and other things. That's cellulose. Now, another one reaction that you did learn, something you did learn. If you take an alcohol and react it with ethylene oxide, your alcohol, ethylene oxide, you get this ether alcohol. Well, guess what? The hydroxyl groups on cellulose, you can do that. And there's a very important product people use. And that's called hydroxyethylcellulose. And here's the molecule. Everybody see that on the right? And we have N, and R can be hydrogens and alcohol, but notice you have CH2, CH2OH. So O, CH2, CH2OH, you take the alcohols and cellulose and treat it with ethylene oxide. You do it in a solvent. Now, the new product, hydroxyethylcellulose, is an amazing thickener. It will take water and make it very thick and make it a good lubricant. Now, one of the interesting places it's used is for personal lubricants. And let's see, they have a nice picture. And no, they don't have the most popular one there. And a personal lubricant, a big company is called KY Lubricant. And a personal lubricant is used for when you're doing something very personal, how should I say, exercise with the opposite sex. And essentially all that is mainly is water and hydroxyethylcellulose, cellulose from plants reacting with ethylene oxide. And that makes that very thick, viscous material because the hydrogen bonds a lot to water. And that's what makes it thick. So next time you're in the supermarket, I don't know about supermarket, well, maybe they probably have it there. But if you happen to be in a big box store like Walmart or that French place, as my friend says, Target, and you go by the personal lubricant area and you'll see KY and all that. Look at the package. Let's hold on. We got time? We got time. All right, everybody, you see KY jelly personal water-based lubricant and has ingredients, water, glycerin, hydroxyethylcellulose. And the rest of those are preservatives and a pH adjustment to sodium hydroxide. Essentially, it's just water and a little glycerin. The glycerin has, you know, the 3 hydroxyl groups. And the thickener to make it that thick gel lubricant is the hydroxyethylcellulose, which you get from ethoxylating cellulose. And that's all I have to do now for carbohydrates right now. Tomorrow, I will go through the carbohydrates practice problem set. Also, tomorrow, we'll do the second to last lab. Oh, that's sad. That's good. Blueberry tea from the Republic of Tea. And tomorrow, because I'll have the time, I haven't told you this, but this whole semester on lab day, if we were face-to-face, we'd have an hour lecture in the lab. And when I do internet during the fall and winter, we go a half hour or so to let you out early. But because summer is so compressed and I want you to do good, I've been spending a lot more face-to-face time on lab days. So you do good, and you have, thank you. But tomorrow we'll probably get out early, but I'll still have time to do some extra practice for test number 4. So show up or watch the video tomorrow. All right, it's new functional group time. Well, actually not new functional group time, new chapter time. And what's that new chapter? Well, I better open it up. Oh, hang in there. All right, everybody see chapter 17, the last chapter. Wow, the semester's almost over. Next week, we'll be done. Wow, where does summer go? Time flies when you're having fun with Dr. White and organic chemistry, including for Dr. White. All right, this new chapter. Jimmy, can you see it? Thank you. All right, it's amino acids, peptides, and proteins. Now, amino acids, what is it? Now, it's a compound that contains both an amino group and a carboxylic acid. I have it written this way, a better way to write it. Is this way. And as you learned for test number 3, this is a carboxylic acid, this is an amine. Now, the true name of amino acids should be amino carboxylic acids, but chemists are lazy and amino carboxylic acids, that's a lot to say. Let's just call it amino acids. So amino acids have both a amine and a carboxylic acid. Now, what I'm talking about in this chapter will not be on test number 4, but will be a couple points, like 10 or so, on the final. So you should learn this. Hint. Now, there can be many things, but let's look at an amino acid. If we look at an amino acid, first of all, an amino acid's always on the alpha carbon, the carbon bonded to the carbonyl, in this case of a carboxylic acid, you have the amino group. Now, if we look at this carbon, the alpha carbon has a hydrogen, carboxylic acid, amino group. When R is not hydrogen, this alpha carbon is chiral. When R is not hydrogen, that's called glycine. I'm not going to ask you to know that, but you should know when R is not hydrogen, amino acids, most of them are chiral. And because they're chiral, they can exist in two forms, an enantiomeric, a right and a left. And that's important. So amino acids can be chiral. You should know that when R is not hydrogen. R can be hydrogen, and glycine is the only amino acid, maybe, I don't know, but it's not chiral. Because you need 4 different groups or 4 different atoms or a combination of 4. Now, switches off, click for this slide. Amino acids contain both an amino group, which is basic, and a carboxylic acid, which is acidic. And depending on the pH, you can get different forms. But if you're at 7, again, switches off, you'll get the proton transferring from the carboxylic acid to the base. Now, if you look on the right here, in the same molecule, you have both a negative charge and a positive charge, net charge 0. That has a name. Now, since I know German, I'm going to pronounce it the German way, Zwitter ion. And you pronounce in German a W like a V in America. And a Zwitter ion comes from the German word zwei, which is 2 ions, and they're in the same molecule. That's why I got the name Zwitter ion. And this switches off. And a Zwitter ion, like I said, is a molecule switched off on this slide too. Click. It has both a positive charge on one atom and negative on another atom in the same molecule. Now, click, switches on big time. Now, if I take 2 amino acids, I can react them together. And let's take a look at the general reaction, which you learned for test number 3. If I take a carboxylic acid and amine, remember R prime and R double prime, one or both can be hydrogen. And therefore, what do you get? An amide. Well, if you look at the amino acid, notice I got fancy. I went to color, full color. Here's my carboxylic acid. Here's my amine. Here's my amide. Now, the amine here doesn't react. This carboxylic acid doesn't, but it's here, and you can react more amino acids. And this calls, now here you're going to see Dr. White's not a protein chemist, but a synthetic organic chemist. I call this bond an amide bond. If you're a protein chemist, you have another name. First of all, let's repeat that. If you start with one amino acid, reacting with itself or a different one, you connect or bond 2 together in this reaction, carboxylic acid, amine, amide. Now, if your switches off, click. But for other classes, you might need to learn this or understand it. So I'm going to give you an introduction. Protein chemists call that bond, the amide bond, a peptide bond. Protein chemists call that a peptide bond. Why they couldn't call that an amide bond? Don't ask me because I don't know. But that's what they call that. All right, now, switch click is still off. A molecule containing 2 or more amino acids in which the amino acids are joined together through an amide bond is called a peptide. Again, I'm not a protein chemist, but if you take 2 or more amino acids and join them together in an amide linkage, otherwise known as a peptide bond, we call that a peptide. Switch is still off. Now, protein is a peptide bond, amide bond, in which you have 50 or more amino acids, and that's called a protein. Why they didn't figure another number, don't know. There's a reason, I just don't know it. So protein, if you look at your skin, protein, it's got 50 or more amino acids joined together in peptide, otherwise known as amide bonds. You've got amides in your skin, your hair, my beard. If you had some protein for lunch, if you're a vegetarian, vegan, if you had soybeans like tofu or something like that, you were consuming amino acids held together by amides. If you were not a vegan, vegetarian, if you had a hot dog, hamburger, steak, or whatever, bologna sandwich, anything with protein from animal source, you were also vegetable source would do it too. You could, but you were consuming amino acids held together by amide. Now, switches on. What happens when you digest a protein? How do you describe when you eat or consume a protein? Well, your stomach and intestines break it down because you have acid and water. And the general reaction, I'll write a little nicer like I did on the past. And you learned this for test number 3. In your stomach, there's HCl and water. And what do you get back? The carboxylic acid you would have used to make that amide plus the amine, salt, not the amine, because you're in acid and amines are bases. And this is the general reaction, how you break down protein when you consume it. Chew your food before you swallow it or you'll choke. And when you swallow it, the proteins, the bonds holding the amino acids together are amides, acid, and water. You get the carboxylic acid and amine salt. However, in a protein, they're connected together. And let's see that. So if you have a peptide or any kind of protein, I have my peptide. No, it's an amide linkage right here. Acid and water. I get back the carboxylic acid plus the amine salt of the amine I would have used to make that. Now, this right here, this happens to be an amine. So in your stomach, it will form the amine salt too. And think about it. Mother Nature has made a polar compound. The amino acid is now polar because they're charged there. And guess what? That makes it more water soluble, easy to transport in your blood. Is she amazing organic chemist or what? And the answer is really amazing. All right, let's talk about what you should need to know for this chapter. And this won't be on test 4, but it will be on the final things you could be asked. Amino acid on the alpha carbon, carbon bonded to the carbonyl carbon of the carboxylic acid, you have an amino group, NH2. You have an R group. When R is not hydrogen, the alpha carbon is chiral, 4 different groups, and the amino acid is also chiral because it's part of the molecule. All right, next thing you should know. If you take two amino acids and react them together, what's the product you get? Uh-oh, I think I'm about ready to, nope, I'm all right. If I was going to lose my memory overload, but what's the general reaction? Carboxylic acid from the first amino acid, an amine from the second one, remember R prime and R double prime can be hydrogen. And you get this amide. Now here's my carboxylic acid. This whole thing right here, I'll call R prime. One of the hydrogens I'll call R double prime. So I'll have my R group on the carboxylic acid, which is really this, carbonyl, my nitrogen, hydrogen, and what's this attached to? This carbon with this R group, and this carboxylic acid still comes along for the ride. And this we call peptide, and that's how you join amino acids. And over here, I can draw more amino acids there, more over here, and it's like a polymer. Amino acid, amino acid, amino acid. So if I were to take and I think you might need an enzyme to help along, but that's another course and I don't teach it. You react 30 amino acids with 30 other, what do you get? Protein. Now, what's the key linkage in a protein that holds the amino acids together? An amide. Again, you should know what is the key functional group that holds amino acids together in a protein, and that's an amide linkage. Now, protein chemists call it a peptide, but I'm not a protein chemist. What does that mean? Leaving my class, you should know what happens when you eat a protein. And the question is, what functional group is in a protein that holds it together? And the functional group is an amide. Therefore, how do you describe with words and a general reaction what happens when you eat a protein? Well, it goes in your stomach, acid, water, hydrolysis of an amide. And how do you show a general reaction? Well, you learned this for test number 3. An amide, you don't have to write this, but I will. Acid, water, you get back the carboxylic acid you would have used to make that amide, plus since you're an acid and amine is basic, you get the amine salt of the amine you would have used. Now, an amino acid, these are connected together in the same molecule. And that's how you do it. And as I show here, you have your amide, you get back the carboxylic acid and the amine salt of the amine. In other words, you get the amine salt of amino acids. And that's how your body breaks down carbohydrate, breaks down protein to be used wherever. Now I'm really beyond my level of expertise. Now, as I said earlier, I've been stealing a lot of time by having you stay later on lab days. Sneaky, aren't I? But that helped you learn better. And what that means is I'm done for today. I've done everything I have to today. I'm done. We did some practice. Tomorrow, listen carefully. Tomorrow, I will go through test number 3 problems and go through the whole test with the answers. It'll be cut out of the video, but always feel free to come to my office hours if you need answers for the test or anything else I can help you with. Next, tomorrow, I will go through the carbohydrates practice problems. Also, tomorrow, I will do my world famous review for the next test, test number 4. And finally, we'll do a really fun lab. And with that, I'm really done for today. I'm gonna let you out early today. First time the whole semester. And with that, gang gesund. I'll see you. Remember tonight, if you need help, stop by my office hour. Bye now.